Okay, so hello. We left off in the last lecture. We were talking about the light reaction of photosynthesis. So for this part of the lecture, we're going to talk about the Calvin-Benson cycle and the dark reaction. The Calvin-Benson cycle and dark reaction are the same thing. And in the Calvin cycle, there is no light. So this is sometimes referred to as the light independent reaction. We made a whole bunch of ATP and NADPH in the light reaction that was made with that electron transport chain. And most of the ATP and NADPH that are produced from the light reaction is going to be used by the Calvin cycle to create what we call, remember we called those G3P molecules. And those are short little 3-carbon molecules. And these 3-carbon molecules work kind of like, think about like a Lego brick. And you would have a whole bunch of Legos, and with those Legos, each Lego is a G3P. If I need glucose, then I can put 2 of those Legos together and get a 6-carbon glucose molecule. Maybe I need lactose. Maybe I need a protein or an amino acid or something to another protein to create microtubules, those sorts of things. All of the different cellular structures and proteins and functional parts of the cell all come from organic molecules. So this G3P is this kind of little Lego brick, and I can put those Lego bricks in any formation that I want to create what it is that I need as a cell. Now, where this G3P comes from is we have to get carbon to make it. So CO2 is what cells are going to take in. So plant cells will absorb CO2 from the environment, and they will create these G3P molecules using the CO2. This is a process known as carbon fixation. And it's the first part of the Calvin cycle. So CO2 incorporation, because we're putting a whole bunch of CO2 molecules together, we're actually incorporating 6 of them, it takes a lot of energy. It requires 18 molecules of ATP and 12 molecules of NADPH. Because it is so energetically unfavorable, this is why the light reaction provides the ATP and NADPH needed for the Calvin-Benson cycle. This is where our plants are becoming what we call autotrophic. Remember, an autotroph is an organism that can use inorganic carbon and convert it into organic carbon for use as an energy molecule. And that's what plants are doing. They're using light energy, so they are called photoautotrophic. They're using light energy to incorporate inorganic carbon. CO2 is a form of inorganic carbon, and converting it into, these are H's, and converting it into organic carbon. But again, that process requires a lot of energy. So 18 ATP and 12 NADPH. We don't want to pull that away from the cellular function, so the light reaction provides the energy that is needed for that. So in this Calvin-Benson cycle, glucose is not made directly. We simply make G3P, and then the G3P molecules, some of them are released into the cell to go become organic molecules. Some of them, however, are going to be recycled in the actual Calvin cycle itself. Very similar to the Krebs cycle, it is a cyclic metabolic pathway, so we're going to have some recycling of some of our products in that pathway so we can begin the process again. So the Calvin-Benson cycle occurs in 3 phases: the carbon fixation phase, the reduction phase, and then the regeneration phase. So in the carbon fixation phase, CO2 is going to get incorporated with a molecule called RUBP, or ribulose bisphosphate. This is done with an enzyme called ribisco. And ribisco is a type of enzyme known as a carboxylase or oxygenase. What this means is ribisco has a very high affinity for CO2, so for carboxyl groups or carboxys, but it also has a high affinity for oxygen. Now, we're not too worried about this oxygen process right now, but that will come into play later in a process known as photorespiration. So remember, Rubisco is both a carboxylase and an oxygenase, has a high affinity for binding either carbon dioxide, CO2, or oxygen molecules. RUBP itself is a 6-carbon molecule. So when we incorporate, when we take a CO2 molecule and we jam it onto an RUBP, it now becomes a 6-carbon molecule. What are G3Ps? G3Ps are 3-carbon molecules. So that 6-carbon intermediate will get split into two 3PGs. In the next phase, we have reduction. In reduction in carbohydrate precursor production, this is where ATP gets used to convert that intermediate into 1,3-bisphosphoglycerate. And then NADPH begins to carry out its redox reactions. It starts adding electrons to G3P and reducing it. And eventually, 6 carbon dioxide molecules are going to be converted into 12 G3P molecules. 2 of those are going to go out to the cell. Now we are assuming, because most of the time this is what happens, those 2 are going to be put out into the cell to become a carbohydrate. In essence, to become a 6-carbon glucose. However, 10 of them are going to be used for regeneration, which brings us to step 3. We need to make some RUBP. So it takes 10 G3Ps to be converted into 6 RUBPs, and this utilizes 6 ATP. This regeneration phase is actually quite large. It's a long process, requires a whole lot of enzymes, a whole lot of energy, and we don't go into a whole lot of detail on that one. So ATP and NADH, where do these 2 products of the light reaction play a role in this whole Calvin cycle? In the fixation stage, the enzyme activity of Rubisco. So Rubisco is an enzyme. It's one of the few that don't end in ASE. The activity of Rubisco of incorporation of CO2 creates the very first intermediate. That was that 3PG. Then we have the reduction stage. The reduction stage takes 12 ATP. That's a lot of ATP. And it converts that 3PG into the 1,3-BPG, or bisphosphoglycerate. This is a 6-carbon molecule, and it's going to get split into 2 3-carbon molecules. We saw that in glycolysis. Remember, in glycolysis, we split glucose, a 6-carbon molecule, into 3-carbon molecules. We then use 12 NADPH to reduce. The NADPH are oxidized themselves, and those electrons are going to reduce that 1,3-bisphosphoglycerate into G3P. That's that carbohydrate precursor. That's going to go out into the cell to go become a glucose molecule. And what it's really doing is it's creating this carbon-hydrogen bond instead of a carbon-oxygen bond. This is inorganic. Carbon-hydrogen is organic. And this is where we see that switch, that conversion of inorganic carbon sources into organic carbon sources. And finally, the regeneration stage uses 6 ATP in that regeneration of RUBP. Because remember, RUBP is getting used up. It's getting used up at the beginning of the Calvin cycle because that RUBP is what binds to the CO2. Those are brought together. Rubisco is just the enzyme that does it. So when we use up this RUBP, means we have to replace it. So it gets replaced during the recycling stage or regeneration stage. So when we do the accounting of the carbons, the Calvin-Benson cycle will actually use a total of 36 carbons. 6 carbons from 6 CO2 molecules are going to get incorporated into 6 RUBP molecules. Now remember, RUBP is a 5-carbon molecule. So I have 6 carbons added to 6 RUBPs. 6 times 5 is 30. So there's my 30 carbon right there. I also have a total, so I end up with a total of 36. I'm then going to split those into 2 for a total of 2 sets of 18, and I end up with 12 3-carbon molecules. I'm still at 36, right? 6 into my 30, right? This is 6 plus 30, so that's my 36. Split into 2 for 2 sets of 18. That 18 times 2 is still 36. 12 times 3 is also still 36. And this gets converted to G3P. We still have 36 total carbons. But 2 of these G3Ps get lost to carbohydrate formation. So that means that 6 carbons get lost, and I have 30 left. So this is minus 6. I'm down to 30. Those 10 G3P molecules get converted into 6 RUBP molecules because 6 times 5 is 30. So we start out with 30. We up 36 because we've added the 6 CO2. We use them, and then we come back to our 30 carbons in the regeneration stage. And these carbons here are what we are trying to get a hold of. So here is our Calvin-Benson cycle. We input 6 CO2 molecules. Rubisco hooks it up with that RUBP. They are combined. Think about the transition step in primary metabolism when we talked about the acetyl-CoA, the formation of acetyl-CoA. It's the same type of thing. RUBP is kind of like a carrier molecule for all of these carbons. Then we have we're now with our 3PG molecule. There are 12 ATP that are used. These 12 ATP are input, right, to give us 12 ADP. Those are going to go back to the light reaction so that ATP synthase can convert them back into ATP. During this conversion, that energy input, we have a rearrangement that occurs, and here's our 1,3-bis phosphoglycerate. So this is that carbon fixation stage, and when we convert this is where we move into phase 2, that reduction and production stage. I'm going to use 12 NADPH, become 12 NADP+. Those are going to go back to the light reaction, and they are going to be reduced again. But when they do this, they release 12 free phosphate groups, and those give us our 12 G3P molecules. 2 of them leave, right? 2 of them leave, and that leaves us with 10 because 12 minus 2 is 10. These 10 are going to be converted back into this RUBP, and so that's where we have our 30 carbons and, of course, our 6 molecules of RUBP. We need these because we need to take in 6 new CO2s to run this cycle all over again. So we lose some down here, but since we recycle, we can gain them back up here. These videos were not working, so we'll try to find one for you and put it in D2L. Now, there are some variations that can occur in photosynthesis, and sometimes different environmental conditions can change or have some kind of effect on either the efficiency of the photosynthetic process or actually in the way that the Calvin cycle actually works at all. And this can be affected by light intensity, temperature, and obviously from the light reaction, water availability plays a really big role. So this is what I had mentioned earlier about photorespiration. Remember, Rubisco functions normally as a carboxylase. So a carboxylase is an enzyme that adds carbon. So when Rubisco hooks up with RUBP, it adds carbon to that RUBP. Remember, it adds 6 of them to 6 RUBP molecules. So these are called C3 plants. C3 plants are the plants that make this particular molecule here. 3PG, that intermediate. That's that molecule that's created when the Calvin-Benson cycle is transitioning from the carbon fixation cycle to that intermediate production cycle. So the problem with C3 plants is that Rubisco also acts as an oxygenase. So in this case, it'll add oxygen to RUBP instead of CO2, and eventually CO2 starts to get released from the cell. Well, the problem is that oxygen doesn't have a carbon attached to it. And the whole point of the Calvin-Benson cycle is to get that carbon. We need the carbon. So this is a process known as photorespiration. And when a plant starts to incorporate oxygen and release CO2, this becomes a very wasteful process. And if there's one thing I know that you guys have learned already about these cells, is this is a no-no in cells. Cells are not wasteful. It's never a good thing when a cell is wasteful. So photorespiration is a process that tends to occur in really hot and dry environments. And this is because of that water loss. So what's happening is, remember, water is absorbed into plants not only through the roots, but let's say we're in an environment where the ground is very dry. Those stomata, they don't want to lose water. So the stomata close, right? And if the stomata close, there's no CO2 coming in. So CO2 is supposed to be moving in through the stomata, and it bounces back. It can't get in. The stomata are closed. The stomata close so they don't lose water. But the problem is that some photosynthesis is still occurring inside of the cell. So let's say this is the inside of the cell. And what's going to start happening is, as what water is left is being split, oxygen begins building up. And when we see this increase in oxygen concentration, eventually enough oxygen is going to make its way to the Calvin-Benson cycle in the stroma, and the rubisco is going to start using that excess oxygen. So by closing the stomata to prevent water loss, when we have, like in a dry, arid, and hot, dry environment, when the plants close their stomata to prevent water loss, in essence, they put themselves into this photorespiration state, because that also closes the stomata to gas exchange. So this photorespiration process is most favored when CO2 concentrations are low and O2 concentrations are high. And that's what's happening when these plants are closing stomata. They're not taking in any CO2, so the CO2 concentrations decrease. They're still carrying out the light reaction with what little bit of water they're receiving from the roots, so in other parts of the plant. So they still have some light reaction going on, and eventually they run out of CO2. And what's left? Rubisco says, well, I don't have enough CO2, I'm going to use O2. And when it starts using oxygen, then the plant starts losing the ability to make G3P. No G3P, no carbohydrate, no autotrophic state. So look at how this is connected, right? Let me start again here. It's dry, which makes stomata close. CO2 levels go down, O2 levels go up. This is due to the light reaction. So we now have rubisco starts using O2, no G3P because I don't have any carbon, no sugars. If I don't have G3P, I can't make sugars. Without sugars, my cell dies. Because without sugars, I don't have primary metabolism. So I don't have enough ATP to make the other cellular processes work. So C3 plants like leaf and white oak, that's what are pictured here, they use the light cycle and Calvin-Benson cycle in what we consider just, your pretty regular average day, every day of the world type of photosynthesis, the kind that we just studied and looked at in this lecture and the last one. But when low CO2 levels start to occur, it causes this photorespiration in plants. And if left in a photorespiration state long enough, the plant will eventually die. But there are two other classes of plants that have evolved ways to avoid photorespiration. And these are called C4 and CAM plants. These guys have developed different alternative ways through the Calvin-Benson cycle, not the light reaction, but in the Calvin-Benson cycle to either prevent water loss or prevent photorespiration from occurring. So corn is what we call a C4 plant. It's also referred to as a monocot. And corn is a really great C4 plant. We grow lots of it here in Florida, believe it or not. And cacti are usually CAM plants. Now, CAM plants, these guys separate processes in their Calvin-Benson cycle, by time. They'll carry out some processes at night and others during the day. Corn does this by separating the two processes in two different kinds of tissue. So one separates through time, the other one separates through location. So let's take a look at C4s. Now, C4 plants make actually a 4-carbon compound that's not found in C3s. And so their carbon fixation process, they actually create something called oxaloacetate. That may sound familiar because that molecule is also what is bound to acetyl-CoA at the very beginning of the Krebs cycle. But this C4 pathway here is called the Hatch-Slack pathway. That's the name of the guys who actually discovered it. Now, what's happening is leaves of these plants, you can see these big long corn stalks, leaves have a 2-cell layer organization. They have mesophyll cells and they have bundle sheath cells. So these right here are the mesophyll, right, these light green. And then the bundle sheath cells are these yellow ones right here. And in the mesophyll, CO2 enters through stomata, right? So the CO2 comes in and it's going to run into mesophyll tissue first. Once it enters in the stomata and it'll get added to a molecule called PEP, phosphoenolpyruvate. And with PEP, which is a carboxylase, we get the formation of a 4-carbon oxaloacetate. So PEP does not promote photorespiration because it doesn't recognize O2. It only recognizes CO2. And if it can't recognize O2, then photorespiration is not going to occur. So you won't see photorespiration occurring in these plants. Now, the bundle sheath cells have take the 4-carbon oxaloacetate and transfer in a way that releases a steady supply of CO2. So what these guys are doing is they're taking CO2 in and they're trapping it in that phosphoenolpyruvate. And they're converting it in that phosphoenolpyruvate, they convert it to a carboxylase and oxaloacetate. In doing that, they can then break off in the bundle sheath cells, get everything to the bundle sheath cells, and then they can start breaking off pieces of that oxaloacetate to create the 3-carbon molecule G3P. And then G3P will be used by the cell to carry out function. In really warm and dry climates, C4 plants are able to preserve water. Think of cacti and some of the plants that live in the desert. And if they can conserve lots of water, they can prevent photorespiration. So in cooler climates, C3 plants will use less energy to fix CO2. But these guys use some of the similar processes to the C4 plants. So here I have C4, right? C4 is I have CO2 entering into a C4 plant in the mesophyll. In the mesophyll, it will hook up with oxaloacetate and become PEP carboxyloacetate. So here's our oxaloacetate 4-carbon molecule here, which then becomes malate. Malate will get broken down and turned into pyruvate. Pyruvate will be oxidized and become PEP. So that's the first part of the cycle. But some of these malates will leave this and get converted into CO2. In CO2, they'll then enter into the Calvin cycle. So in CAM plants, we have spatial separation of the Calvin cycle from another cycle up here. So this is the PEP cycle. This is the Calvin-Benson cycle down here. So CAM plants are from, CAM stands for crassylation acid metabolism. That's what CAM stands for. And some C4 plants have evolved or changed their processes using time. CAM plants will only open their stomata at night, and that's because it's much cooler. So in the cool night air, they're less likely to lose water through the stomata. When the stomata is opened at night, CO2 will enter it and is immediately converted to that malate sugar. And stomata will start to close during the day when the light comes up, and they will close in order to conserve their water. And malate is going to then be broken down by CO2 and is going to be part of the driving force in the Calvin-Benson cycle. So here we just have a comparison of C4 and CAM. C4 over here is separating things spatially, right? We have this type of cell layer and then our bundle sheaths. So we have a separation of these two processes. Over here in the mesophyll stage, here in CAM plants, we're not separating spatially, we're separating by time. At night, this cycle is happening. During the day, this cycle is happening. What these guys are doing is they're making molecules that can break off pieces of CO2. So they're later during the day, they're allowing this Calvin cycle to occur to create all those G3P molecules, but their stomata are closed. They're not taking in any CO2. At night, they stop their Calvin-Benson cycle and they just allow the C4 cycle to occur. And in the C4 cycle, they're making that oxaloacetate, and oxaloacetate is used to store CO2. That's what's happening. So we have a C4 cycle in both of these processes here. Just one of them, the C4 cycle is occurring in different tissue. In the other one, the C4 cycle is occurring at a different time. So these plants have evolved really amazing ways to prevent photorespiration. They're not going to have photorespiration occur because they're not producing high levels of oxygen. The oxygen concentration is being kept fairly low. And at night, when these guys, their stomata open, that oxygen will be released out into the air and CO2 is allowed to be absorbed. So that's the end of the photosynthesis lecture. Your essay questions are already available, and I already posted the lecture from the other day. Good luck on your exam. Remember, it's going to be due by Sunday, and have a great evening.